the periphery, they are smaller, and inside they are larger. So you need to see that the peripheral bundles are smaller and the inner bundles are larger. Coming to the special feature in the monocot stem, that is the phloem parenchyma we have seen in this. See, this is the transverse section that we cut in a stem. In the first picture you can see the transverse that is cut like this. When we take one part here, the single part we take under microscope and we study, then the picture appears to be like this. So we see the first, what layer do we see? We first see the epidermis. Okay. After epidermis we have cortex. In cortex we have three layers, that is hypodermis, general cortex and endodermis. After cortex we have pericycle. Okay, and the pericycle lies in between the endodermis and phloem. After pericycle we have medullary rays. Medullary rays are made of parenchymatous cells and they lies in between the vascular bundles. After medullary rays we have vascular bundles. These vascular bundles have radial arrangement, and each bundle has a conjoint arrangement of xylem and phloem, where they both are arranged together. In between xylem and phloem we have cambium. We have already studied in the conjoint arrangement, when there is a cambium, the phloem lies outside and the xylem lies inside. Phloem is at the outer side of the vascular bundle and the xylem lies in inner side of vascular bundle. This is the point we have studied in the morphology of flowering plants. After vascular bundles, the next part that we see is our pith. So these are the 6 layers present in a dicot stem. The other important point in pericycle, the pericycle is arranged in semilunar patches of sclerenchyma. As the cells of sclerenchyma are present, the major function of this pericycle is mechanical support. This helps in mechanical support. So now we are done with the dicot stem. After this we will go to the monocot stem. Okay, even in the monocot stem all the things appear the same. We will write all the layers of monocot stem now. Okay, in the monocot stem, the very first layer that we have is the epidermis. What is epidermis? Epidermis is the outermost protective layer and it is covered by cuticle to prevent the water loss. After epidermis, next we have hypodermis. In the cortex we don't have 3 zones, we just have a single zone of hypodermis, and this hypodermis is made up of sclerenchymatous cells. These sclerenchymatous cells, they provide support. After hypodermis, the next layer we have is our ground tissue. So in ground tissue is present, this is an entire tissue made of parenchymatous tissue. Okay, ground tissue is totally made up of parenchymatous cells. In a monocot stem, the major thing you need to remember is the cortex, endodermis, pericycle and pith. All these 4 are not distinct. We can't differentiate between all these 4 are not distinct, and all these together are seen under the ground tissue. After hypodermis, the cortex, endodermis, pericycle, pith, they are not distinct and all are grouped together and studied under ground tissue. So the function of ground tissue is here, its storage. So we are seeing epidermis, hypodermis and then ground tissue. After ground tissue, the 4th part we have is the vascular bundles. Coming to the vascular bundles, in vascular bundles we have numerous and scattered vascular bundles are seen. We see scattered vascular bundles in the ground tissue. Okay, it's like here we have too many parenchymatous cells. This forms our ground tissue, and in the ground tissue we have these scattered vascular bundles present somewhere. Okay, the vascular bundles are scattered in between the ground tissue, and each vascular bundle we see, it is a conjoint bundle, that is the xylem and phloem are arranged like this. And the other thing is that they are closed. As we are seeing a monocot plant, the cambium is not present, the secondary growth is not seen, so it is closed. Other thing is these vascular bundles are surrounded by sclerenchymatous bundle sheet. We have vascular bundles here. These vascular bundles are surrounded like this by a sclerenchymatous sheet. Okay, vascular bundles are surrounded by a sclerenchymatous sheet. Okay, so other thing to remember is, as we have the scattered vascular bundles here, the vascular bundles at the periphery are smaller. You can see in the picture the vascular bundles present here in the periphery, they are smaller, and inside they are larger. So you need to see that the peripheral bundles are smaller and the inner bundles are larger. Coming to the special feature in the monocot stem, that is the phloem parenchyma is absent. Phloem parenchyma is absent. Other thing is the water containing cavities are present in vascular bundles. The water containing cavities are present in the vascular bundles. What are the two major differences here? The phloem parenchyma is absent and the water containing vascular cavities are present in the vascular bundles. What are the layers we have seen? See, this is a transverse section, and when we look at some part here, it looks like this. We have the outer epidermis, and inner to epidermis we have a hypodermis layer made of sclerenchyma. Inside the hypodermis we have ground tissue. Vascular bundles are scattered. We have scattered vascular bundles in the ground tissue, and the vascular bundles are surrounded by a sclerenchymatous sheet. So this is the structure of a monocot root. Now we will see the key differences between a monocot stem and a dicot stem. So first, looking at a dicot stem and a monocot stem. In the dicot stem, the vascular bundles are in ring, like they are in the radial arrangement. They have open bundles, that is cambium is present, so the secondary growth is seen in them. Coming to a monocot stem, we have the scattered vascular bundles, and these are closed as it's monocot plant and they have no secondary growth. The vascular bundles are covered by sclerenchymatous sheath. They have no phloem parenchyma. Okay, and there is no distinct cortex with pericycle and endodermis. We only have a group of parenchymatous cells that are arranged in the ground tissue. In the ground tissue itself, we find the scattered vascular bundles. Okay. So now we are completed with our monocot and dicot stem also. I'll write the differences properly now. 1 second. Coming to the differences of a dicot stem and a monocot stem. We'll start with our first layer. The first layer we studied is the epidermis. Okay, the epidermis of a dicot stem is present with cuticle to prevent the water loss, and it may also have trichomes, hair-like structures. In monocot also epidermis is present and cuticle is also seen. There are no much differences in the epidermis. Coming to the hypodermis. Okay, the hypodermis in dicot is made up of collenchymatous cells, and whereas in monocot it is made up of sclerenchymatous cells. And then coming to the cortex of dicot stem has 3 zones and is well differentiated. Coming to the monocot, the cortex is not differentiated. In fact, a separate cortex is only not present. The cortex is not distinct, it's included in the ground tissue itself. Next, coming to the endodermis, here the endodermis is present and is called as starch sheath, as the cells are rich in starch. Here the endodermis is absent in the monocot stem. Next, coming to the pericycle of dicot.
Students keep writing notes, while doubts stay uncleared. Hours of self-study, yet biology still feels confusing. Because sometimes you don't need more lectures, you need personal guidance. At Crack Mentors, students get live personalized online one-on-one biology classes with dedicated faculty support, personal doubt solving, and focused improvement strategy.